Okay, so welcome to this next video in which we are discussing type 1 activation of endothelial cells. So far we've discussed that when you stimulate an endothelial cell with a pro-inflammatory molecule that's coupled to the GQ pathway, such as histamine, which will bind the H1 receptor and activate the GQ pathway, then we've firstly seen that that will lead to the production of the two vasodilatory molecules, prostacyclin and nitric oxide, and now we're seeing how it leads to contraction of the endothelial cells. So we've seen that by activating the alpha-Q GTP subunit, the ligand binding to the G protein coupled receptor will cause release of calcium from the endoplasmic reticulum stores, and that this will bind to calmodulin to form calcium calmodulin complexes, which will then bind and activate the myosin light chain kinase. Okay, the myosin light chain kinase will then phosphorylate myosin light chain here. However, we discussed that that's pointless if you don't inactivate the myosin light chain phosphatase, because if myosin light chain phosphatase is not inactivated, it will just remove these phosphate groups instantly off myosin light chain before it can do anything. So then we saw that actually the beta-gamma subunit of the heterotrimeric G protein is important, that it binds to and activates the RAS homology guanine nucleotide exchange factor, ROGEF, which then activates the G protein RAS homology, or Rho, and takes the GDP that's bound to it off and binds GTP instead to make Rho GTP. Rho GTP then activates Rho-dependent kinase, which then phosphorylates and inactivates myosin light chain phosphatase, and this means that you've got an activation of myosin light chain kinase and an inactivation of myosin light chain phosphatase, so you overall get myosin light chain being phosphorylated and staying phosphorylated, and that this somehow then leads to the contraction of the actin filaments which are attached to the cytoplasmic domains of the proteins involved in both the tight junctions and the adherens junctions between neighbouring endothelial cells. And that this then leads to the two endothelial cells pulling apart and opening a little gap in between them. And this is known as endothelial cell contraction, or just endothelial contraction. Okay. And it opens up these holes in between the endothelial cells. Now, what's the significance of these holes? Well, it's really, really important in the venules and the capillaries. So you get a huge amount of these holes being formed in both the capillaries, but more importantly in these venules. Basically, all of these gaps are now going to open. Now, what does this mean? Well, it means that contents from the blood can start to leak out into the interstitial space where the Staphylococcus aureus is. So you're going to allow all sorts of proteins that are within the blood to leave the blood and go into the interstitial space that would not usually be able to get into the interstitial space. And these proteins are going to help fight the Staphylococcus aureus. So we're not going to go into detail of these proteins here, we'll do that in other videos. But two examples of very important proteins that are going to come out are, you're going to get complement proteins coming out, which are very important in attacking the Staphylococcus aureus directly, and also you're going to get coagulation factors coming out. And these coagulation factors will start to set off the coagulation cascade, because as soon as coagulation factors leave the bloodstream, they come into contact with collagen and tissue factor, which then sets off the intrinsic and extrinsic coagulation cascades and leads to fibrinogen being converted into fibrin, which is then assembled into fibrin strands. So what on earth is the point of that? Well, basically you're going to get a meshwork of fibrin being formed in the interstitial space, and this will basically trap the Staphylococcus aureus in. So it can't move, it can't spread basically, because a fibrin meshwork is just penning it in. Okay, so both of these protein systems are extremely important, and when you open these gaps between the endothelial cells, these are going to get out into the interstitial space where the Staphylococcus aureus is. Okay, right, so that's the importance of the contraction of the endothelial cells. Okay, right, and that's happening mainly in the venules and the capillaries. Now, this increased fluid that comes out of these now very leaky venules and capillaries, this is what's known as an inflammatory exudate. So where should I write this word? I'm going to put it here because of the lack of space up there. So it's known as an inflammatory exudate, or merely just as an exudate. Okay? And this causes the volume of the interstitial fluid to go up considerably, and this is what leads to the swelling, the tumor of the 5 pillars of inflammation. Okay, so it's the opening of these gaps in between the endothelial cells due to the endothelial contraction that causes the swelling of the inflamed area. Okay, so we're now going to move on to the final portion of the activation, well, the type 1 activation of endothelial cell pathway, which is that we're going to start grabbing on to blood cells, to white blood cells in the lumen of the blood vessel, and we want them to be moved into the interstitial space. So far what we've done is we've got proteins into the interstitial space. We've got complement proteins and coagulation cascade proteins, as well as a bunch of other proteins that are less famous than those two, okay, that are going to help fight the Staphylococcus aureus. However, these holes in the side of the blood vessel are not big enough for whole cells to go through. White blood cells are not going to get through these little holes that you've created. But we want white blood cells to get into the area because they are there. They are cells that are specifically designed to attack things like this, to deal with problems like Staphylococcus aureus. Okay, so we want to recruit leukocytes, and leukocytes is just the fancy word for a white blood cell. Okay, now the first type of leukocyte that is recruited by endothelial cells that have undergone type 1 activation is known as a neutrophil. Okay, so neutrophils are a major type of leukocyte within the bloodstream. Okay, and they are, it's fair to say, that one of the dominant forms. They are quite a cheap cell to make, basically. It's like they're not your best fighters, but you've got a lot of them. They're kind of like the pawns in chess. You'll send them in, and you'll reserve the better pieces for later. You'll use them later. So they're quite a cheap cell. You've got a lot of them, and you're just going to pile them in there initially. And they're the first move, basically. Okay, so we're now going to look at how type 1 activated endothelial cells are going to move neutrophils from the lumen of the blood vessels into the affected interstitial space. So before I just do that, I'm just going to describe to you what a picture of a neutrophil looks like. So neutrophils are often also known by the name polymorphonuclear cells. So polymorpho, and then it's supposed to be all one word, but never mind, nuclear cells, okay? Or PMNs for short, polymorphonuclear cells, or just PMNs. People often do abbreviate them just to PMNs. And the reason you can abbreviate them to PMNs without the cells on the end is because you can also call them polymorphonucleocytes, okay? And that's how you can turn this into one word, basically. You can combine nuclear cells into the word nucleocytes, and that's where PMNs comes from. Okay, so polymorphonucleocytes. Now, why are they called polymorphonucleocytes? Well, because the structure of their nucleus is very odd. Basically, they have multi-lobed nuclei, and the typical archetypal number that's generally drawn is 3. But generally, if you actually look in histology, I think you generally find 4 usually. But the archetypal textbook number is 3 lobes of the nucleus. Okay, so here is their nucleus that's got this very odd lobed shape like this, and this is why they're called polymorphonucleosides. And the reason it's polymorpho is because if you look at a nucleoside, they don't have a fixed number of lobes. It's not always 3. Sometimes it'll be 2, sometimes it'll be 3, sometimes it'll be 4, sometimes it'll be 5. So they have loads of different shaped nucleus, and this is why they're called polymorphonuclea or polymorphonucleosides, because many is what poly means, morpho means shape or morphology, and then nucleus, so they have many different shaped nuclei, okay? So that's the logic in their name. Okay, so what we now want to look at is how are type 1 endothelial cells going to recruit these guys to the site of infection. Okay, and I should probably, just before we start that, tell you what the neutrophils will do once they're there. Basically, they are phagocytes. So once they get there, they will start phagocytosing the Staphylococcus aureus. They'll start taking them into their cytoplasm. So I'll show this. Here's a little Staphylococcus aureus. What they'll do is they'll form a little invagination known as a phagosome, and they'll take the Staphylococcus aureus into a phagosome within their cytoplasm, and they'll then break the Staphylococcus aureus down. Okay, so that's what's meant by phagocyte. And this process of taking a bacterial cell up into your cytoplasm and breaking it down is what's known as phagocytosis. Okay, and to make everything clearer, I will colour in the Staphylococcus aureus in blue here. Okay, so this shows the multiple steps of the process. It shows the invagination of a phagosome, and I suppose I should write that down as well. Phagosome. Okay, and then it goes into a phagosome, and then that phagosome will fuse with lysosomes, and the Staphylococcus aureus will be digested. Okay, so how do we start recruiting these leukocytes to the site of infection? How do we get them out of the blood and into the interstitial fluid? Because they can't usually do that. They can't usually get across the endothelium. Okay, so when the endothelial cells undergo type 1 activation, it's going to allow these endothelial cells to recruit the neutrophils and move them across the membrane. Okay, so what happens? Well, this also involves myosin light chain, basically. Okay, so endothelial cells which have not undergone type 1 activation do not have receptors for binding to proteins on neutrophils on them. Okay, so let me say that again. Basically, what's going to happen, in order to get these neutrophils to come across the endothelium and into the interstitial fluid, we are going to put on our surface a receptor for a protein that is on the surface of the neutrophils. Okay, so let me tell you what the name of this protein that's on the surface of neutrophils is called. So on the surface of the neutrophils, you have a protein known as PSGL1. Okay, so this is PSGL1. Now, what does this stand for? This stands for P-selectin, so that's the PS, and then glycoprotein is the G, glycoprotein, and then the L is a ligand. So it stands for P-selectin glycoprotein ligand 1. Okay, so on the surface of the neutrophils, you have this protein known as P-selectin glycoprotein ligand 1, or PSGL1. And that's only just visible, that was lucky. Right, so what you're going to do is, in order to catch the neutrophils as they're moving through the blood vessel, you're going to put on the surface of these type 1 endothelial cells a receptor for the PSGL1. And the receptor for PSGL1 is a protein known as P-selectin. And it's actually a glycoprotein, funnily enough. So now, this name PSGL1 seems perfectly sensible. P-selectin glycoprotein ligand 1. So, all we need to do is put on our surface this P-selectin. It's slightly more complicated than that. I'll show you that actually you need two things, but P-selectin is one of the main ones. So, what happens? Well, basically, I told you that earlier on that type 1 activation of endothelial cells does not involve any change in gene expression. That takes too long. This is an extremely rapid response, basically, to histamine. Type 2 activation is going to involve a change in gene expression, but we're not there yet. We're on type 1 activation. Okay, so that must mean that the endothelial cells are not going to produce the P-selectin. Instead, they must already have the P-selectin. Okay, and indeed they do. Basically, they have P-selectin, but it is not yet in their apical membrane, so it is not yet visible to the neutrophils. Instead, it is kept securely inside vesicles within the cytoplasm of the endothelial cells. Okay, so this protein here in this vesicle, which I will colour in vivid purple. Okay, so this protein in vivid purple, this is the protein, or rather the glycoprotein, P-selectin. Okay, so these vesicles that are inside the cytoplasm of the endothelial cell that has not yet undergone type 1 activation, so this is what's present in the inactive endothelial cell. Basically, these vesicles have a very important name. They're so important they've been given their own name. So they're known as Weibel or Weibel Palade bodies. Okay, so Weibel Palade bodies. And these are often abbreviated to WPBs. So this is a WPB. So the name for a vesicle within the cytoplasm of an endothelial cell which has P-selectin within it is a Weibel Palade body. Okay, now, so in order to catch endothelial cells, all we need to do is exocytose these Weibel Palade bodies so that the P-selectin is now on the apical surface of these endothelial cells and is exposed to the PSGL1 so that the neutrophils can now stick to the P-selectin and therefore will have the tethering of the neutrophils to the apical membrane of the endothelial cells. So at least that's a step forward. Okay, so what's going to happen is that when the endothelial cell is activated, we know that that's going to activate the phosphorylation of myosin light chain. Okay, and the myosin light chain being phosphorylated is also extremely important in the exocytosis of these Weibel Palade bodies. So when you phosphorylate myosin light chain, that causes the exocytosis of the Weibel Palade bodies, and that leads to the P-selectin being exposed now on the apical membrane of these endothelial cells. Okay, so let me go through this again, summarize this process again. Okay, so in the inactive endothelial cell, you have P-selectin sequestered not on the apical membrane of the endothelial cell, but instead in these Weibel Palade bodies, these WPBs, okay, which are vesicles within the cytoplasm of the endothelial cell. Okay, now when you activate the G protein coupled receptor that's GQ coupled, such as the H1 receptor by histamine, then we know that that's going to lead to the activation of two things, the alpha Q subunit and also the beta gamma subunit. And the alpha Q subunit leads to the calcium signal, so it leads to the release of calcium into the cytoplasm of the cell via the production of IP3. And that calcium then binds to calmodulin to form calcium-calmodulin complexes, which activates the myosin light chain kinase, which phosphorylates myosin light chain to myosin light chain with a phosphate group on it. However, we saw that that was pointless if you didn't then stop the activity of myosin light chain phosphatase, because if you didn't inhibit myosin light chain phosphatase, it would instantly remove the phosphate group of myosin light chain. So what we also did is we had the beta-gamma subunit activating the Ras homology guanine nucleotide exchange factor, ROGEF, which then activated the G protein RAS homology, Rho, by ripping the GDP off it and binding GTP to it instead. You've then got Rho GTP, which activates Rho-dependent kinase, and then Rho-dependent kinase inactivates, phosphorylates and inactivates myosin light chain phosphatase. So that's how you achieve inactivation of myosin light chain phosphatase. So you've now got activation of myosin light chain kinase, inactivation of myosin light chain phosphatase. You get myosin light chain phosphorylated, and myosin light chain phosphorylated myosin light chain then leads to the exocytosis of the viable palade bodies, so that the P-selectin is now on the apical surface of the endothelial cells. Now, what we'll see in the next video is that P-selectin is not quite enough to cause the tethering of these neutrophils. It's a powerful it's a powerful part of it, but there's actually another thing that also helps tether these endothelial cells, and we'll discuss that in the next video. This is where we're going to go back to the lysophosphatidylcholine, which I said we would come back to.